Hi everyone, am I audible? Hello? Hello? Am I audible? So, Vanakkam everyone, welcome to Unacademy NEET English. I'm your biology educator Ambika Sharma, and today we are going to continue molecular basis of inheritance, and we will complete DNA replication and then we will go for the transcription. So these two topics we are going to complete today. Okay, so excited for the new topics? Yes, excited for the new topics? And do let me know in the chat section, am I perfectly audible and visible to you all or not? Hi Subhashini, Trisha, Purple. Yes, everyone. Guys, what is happening these days? You people, your number is very less, right? The energy is very less. You requested this chapter. We are going very slow. We are going in detail of the topics. We are discussing them in detail, but still, the response is very lame. What is happening these days? Good morning. Good morning. Huh? So don't you like the morning slot? You want classes in the evening? Huh? Do you want classes in the evening or the morning is fine? I think it's perfect. You're starting your day with the genetics. Exactly, you really need to be energetic. Be a support, guys. Quickly hit the like button. If you're new to our channel, hit the subscribe button and send this link to your friends. Share it with your friends and let them know that you are attending amazing classes of the molecular basis of inheritance. Morning is perfect. Yeah, even I feel the same. Okay, my classes I'm already taking, Manpreet, I'm already taking Avengers batch on our Plus platform. Wow, very nice. Students are from Meghalaya as well. That's great. That's good. So, be energetic. Take out your pen and paper. Keep your book with yourself. And whenever I will tell you that this point is very important, please highlight it in your book. Okay, highlight it in your book. Right. So let's start the class, guys. Let's start the molecular basis of inheritance. This is a topic, right, that we are doing from last, I think, 2 to 3 sessions, and soon we will finish it. And I will try my best. I'm actually planning to take a marathon session on Sunday. Okay, and I will finish this chapter in one go then. Okay, by this week, let's finish molecular basis of inheritance. Let's finish body fluid and circulation. And from next week, we'll start the evolution. Okay, we'll try to keep, I will try to increase the duration of the classes. Don't worry about that. Fine, we'll try to increase the duration of the classes so that, we can discuss it properly in 2 or 3 parts so that we can finish the chapters. Don't worry, I'm planning about that too. Yeah, I used to cover everything in the marathon. So let's not waste time. Let's go for it, guys. So hit the like button and be attentive. So let's start with the revision. So in the last class, we started the DNA replication. So DNA replication is going to follow which mode of replication? Yes, when it comes to the DNA replication that we are studying, so which mode of DNA replication it is? Tell me quickly, which mode of DNA replication it is? Very simple. It is the semi-conservative mode, or it is the semi-conservative model of DNA replication, isn't it? Isn't it? Messelson and Stahel, in the case of prokaryotes, in the E. coli, they provided the experimental proof for the semi-conservative model. But when you talk about the eukaryotes, like the plants, the faba beans, then Taylor and his colleague, they experimentally proved it. That's what we have covered in the last class. So now you know that in the S phase, okay, in the S phase of the interphase, the DNA replication will occur, and we need certain enzymes for that. So to start with, helicase, single-stranded binding proteins, right, topoisomerase, and yes, I checked the comments as well. Many students are there, right? They are writing the wonderful comments. Thank you so much, everyone. You are even mentioning your homework. That's a wonderful thing. Right? May God bless you all. May God bless you all. If you are following the instructions, if you are, sincere, if you are putting efforts, definitely you will get selected in your dream medical college. Okay, so these are the enzymes. So helicase, SSBP, topoisomerase, where we discuss DNA gyrase in prokaryotes. Right, bache? Other than that, primase is there. Okay, and then comes your DNA polymerase. So that's what we have discussed in the last class. So now let's quickly revise it, quickly recall it, and then we will proceed. Fine, bache? So you know that in our syllabus, mainly we discuss about the replication of the bacteria. And which bacteria it is? It is E. coli. So in our syllabus, majorly we talk about the prokaryotic DNA replication. So as I said, there are, right, certain things are different in prokaryotic DNA replication and eukaryotic DNA replication, but almost the process is same. Right, bache? Almost the process is same. Clear, bache? Okay. Okay, so here you know which DNA do we have? It is the circular DNA. So, bache, replication cannot start from any point. It cannot start randomly. It's not like that. Replication is going to start from a particular point, and that site of replication is known as, yes, everyone in the chat section, do let me know the answer of this question. The site of replication, exactly, bache, it is your origin of replication. The point from where the replication starts is origin of replication, and in the case of E. coli, it is ori C. Clear, bache? In the case of E. coli, it is ori C, right? This is the site of replication. And again, let me remind you this thing, bache. In the case of bacteria, in the case of prokaryotes, it is going to be monoreplicant. There is only one point from where the replication will start. But in the case of eukaryotes, right, in the case of eukaryotes, there will be many origin of replication. That is going to be polyreplicant. Okay, it is going to be polyreplicant. That is something very important, right? So now in the case of E. coli, because the DNA is what? It is circular. What type of DNA is there? It is circular, and there is one point from where the replication will start. And what we used to call it? As I said, it's ori C. Now, there are certain proteins, right, present here in the cytoplasm, and even if you are talking about the eukaryotes, in their nucleus, there are certain proteins, which Ashok, that will detect this origin of replication. In the case of eukaryotes, because the DNA is linear, so there are many origin of replication, and there are proteins which will detect it. Now what is going to happen? Our enzyme, our unbindase, this helicase will play its role. Right? It is also known as unbindase. We have covered it in detail in the last class. So you guys can check that as well. Okay. So the unbindase, the helicase, okay, it will come here. Why? Because it is going to break that hydrogen bonds. You know that in the DNA, we have the complementary base pairing. You know that if two strands are there, they are complement to each other. If one is having adenine, another will have thymine. If one is having cytosine, another will have guanine. So that's how it works, isn't it? That's how it works. You know it very well, students, right? You know it very well, isn't it? So now the point is that if I want to make the new dot strands, we know it is the semi-conservative model of DNA replication. So we have to open up our DNA so that we can copy that information, right? So that we can copy that information. So you know about it, isn't it? You know about it. Like this is the double-stranded DNA. This is the double-stranded DNA. You know that. So if we want to copy that information, we need to open up this double-stranded DNA. What we need to do? We need to open up this double-stranded DNA. And which enzyme is going to play the role here, bache? It is your helicase, right? It is your helicase. Is that clear? Yes, everyone. Is that clear? It is your helicase. Clear, bache? Clear? And when it comes to the helicase, always remember that it requires energy, right? And by ATP hydrolysis, it is capable of breaking these hydrogen bonds. Okay, and the cofactor for this enzyme is your magnesium ion. Okay, that is your Mg2 positive. That is your Ng2 positive. So now it is the double stranded DNA. My helicase is breaking the bond, but it will do it very slowly. Remember in the last class I gave you the example, right, that it is going to open the DNA strand but very slowly, so that we can get this Y-shaped replication fork, right? And why is it so, bache? Because it is a what? It is energetically very expensive process. So helicase, when it is breaking the hydrogen bond, it requires energy. So obviously slowly it will open up the DNA strand. Fine, it will open up the DNA strand. Now these DNA strands, see, these DNA strands, these separated DNA strands, these single strands, they act like what? They act like the template. They will provide us information to copy the DNA. Clear, bache? They will provide us information to make the new daughter strand here. Is that clear? Is that clear? And moreover, Dushyant, right, these single strands, they can re-anneal, they can join again, they can again get coiled. Okay, they can again get coiled. So that's why we have single stranded binding proteins. They will bind to these strands of DNA. They will not allow these single strands to get coiled again. Is that clear? Is that clear? That's what you need to keep in your mind, and that's what we have studied in the last class. So in the E. coli, because this replication is bidirectional. What type of replication is there, bache? It is bidirectional. Okay, it is bidirectional. So here at this particular point, what will you see? You are going to see the replication bubble. Okay, you are going to see the replication bubble. So replication bubble is having two Y-shaped replication forks. Is that clear? It is having two Y-shaped replication forks. So this is what we have covered in the last class. So in the last class, bache, I also told you about the topoisomerase, right? I have seen that many students, they have given the answer in the chat section, in the comment section. Very good. So the next enzyme here is Subhashini, topoisomerase. So now you know. See, now you know. See, if something is helically coiled like this, so if you are trying to open it, some tension will be created. We have experienced it in the case of ropes as well. If we are trying to open up, right, if we are trying to open up something helically coiled, I know a tension will be created. The supercoilings can be generated. And to remove such supercoilings, we have topoisomerase. Because see, imagine, helicase is moving like this. Just imagine, okay? Helicase is moving like this. But here, right, let's say if there are some coilings, so how that helicase will open up DNA strands? It is not possible for helicase to do so, isn't it? It is not possible for helicase to do so, to open up this DNA strand. Then what is the case? What is the point? What is the scenario here? We need another enzyme, that is your topoisomerase. Okay, that is your topoisomerase. This is the enzyme that we need, isn't it? This is the enzyme that we need. So this topoisomerase, it will remove supercoilings, right? It will remove that supercoilings, and yes, even it requires ATP. Clear? Even it requires ATP. Is that clear? Is that clear? Right, Subhashini, if you don't understand, bache, okay. You are saying that it is meant for supercoiling. Great, bache, great. So see, this is how. So see, you're trying to open it. Some coilings are there, na? Here also you guys can see. So topoisomerase is going to remove that coiling. So as I said, in the case of topoisomerase, you have two classes, class 1 topoisomerase, class 2 topoisomerase, and this class 2 topoisomerase can cut both the strands of DNA, then they can remove that coiling and will again join DNA. And class 1 topoisomerase, it can cut only one strand of DNA. That's what we have studied in the last class as well, bache. As of now, whatever I am telling you now, we have covered it in the last class, right? Because I just want you to be in that flow. That's why I'm explaining it again. So up to this part, all clear? Yes? Up to this part, all clear? Sure? Sure? Okay, very good, Nandini. Excellent, excellent. So, in the case of prokaryotes, you talk about the DNA gyrase. Okay, and that is also a topoisomerase. It is the first topoisomerase discovered in prokaryotes, that is DNA gyrase, which is a class 2 type of topoisomerase. Right? So, its function is to remove the positive supercoiling, but it introduces negative supercoiling. Right? So it removes the positive supercoiling, but it introduces the negative supercoiling. So that's what you need to keep in your mind. Is that clear? That's what you need to keep in your mind. So this is about the topoisomerase. So bache, in the last class, we were also discussing the polymerases, isn't it? We were also discussing the polymerases. I hope you remember. We have DNA polymerase 1. We have DNA polymerase 2. We have DNA polymerase 3. Isn't it? So DNA polymerase 1 is there, DNA polymerase 2 is there, DNA polymerase 3 is there. That's what we have. So, but this DNA polymerase 1 is also known as Kornberg enzyme. It was discovered by Kornberg, and this enzyme was used to make the DNA, right, even in the lab, right? This enzyme was used to make the DNA even in the lab. So, it is the DNA polymerase 1. Which enzyme it is? It is the DNA polymerase 1. Now, when it comes to the DNA replication, understand this part. What are you going to do? Okay, what are you going to do in DNA replication? Wait. But ultimately you are going to synthesize a new strand. You are going to form a new strand. And how that new strand will form? When you will add the nucleotides, right? When you will add the nucleotides, and then you will form that strand. So ultimately the polymerization will be there, Nandini. Ultimately the polymerization will be there, and that polymerization will form the new strand, isn't it? That polymerization will form the new strand, isn't it? Isn't it? That's what you know. That's what you know, isn't it? So of course, for the synthesis of new strand, it is obvious that we need the enzyme which is having the polymerizing activity. So that's why it is DNA polymerase. Okay, that's why it is DNA polymerase. But the main replicating enzyme is this one, DNA polymerase 3, also known as replicase. DNA polymerase 3, also known as replicase. Clear, bache? So when it is DNA polymerase 1, it is having the activity of exonuclease. Right? It is having the activity of what? Exonuclease. So 5' to 3. So yes. 3' to 5' exonuclease activity. I will explain it today, that what is the meaning of this type of activity. Okay, but it is also unique. It is also having 5' to 3' exonuclease activity. That is something unique related to this DNA polymerase 1. That it is also having 5' to 3' exonuclease activity. Just keep it in your mind. Okay, okay. And yes, even this enzyme is also having the polymerase activity. 5' to 3' polymerase activity is there. Okay, 5' to 3' polymerase activity is there. You should know about these enzymes. Okay, now come to the DNA polymerase 2, which is least active. It is least active. It is also having 3' to 5' exonuclease activity. So if DNA polymerase 1 and 3, if they are absent, then your DNA polymerase 2 is going to work. Clear, bache? Then your DNA polymerase 2 is going to work. Otherwise, this enzyme, yes, bache, otherwise this enzyme is least active. Is that clear, Ashok? This enzyme is least active. Clear, bache? Clear, bache? So its polymerizing speed is very less. It is also having polymerase activity, but this polymerizing speed is very less, bache. Okay, this polymerizing speed is very less. You can say that near about 50 nucleotides per minute. right? So it's very, very, very slow. So these two enzymes mainly, they play a role in DNA repair. Okay, now come to the DNA polymerase 3. So it is having 5' to 3' polymerase activity, of course. So it is the main replicating enzyme. It is the one which will form our new strands. Is that clear? It is the one which will form our new strands. Is that clear? And its speed is too much, bache. Imagine about 2000 base pairs per second, right? Around 2000 base pairs per second. Just imagine. Just imagine. DNA polymerase 2 is adding near about 50 nucleotides per minute. But when you talk about this DNA polymerase 3, imagine your 2000 base pairs per second. So this enzyme, right? This enzyme, this DNA polymerase 3, okay, this enzyme is fast as well. Moreover, it is accurate as well. Okay, so the mutation, the chances of mutations are very, very, very less, and that's what we need when we are replicating our DNA. That's why this is the one which is our main replicating enzyme, and also this is also having 3' to 5' exonuclease activity. Okay, it is also having 3' to 5' exonuclease activity. Okay, now I will tell you the role of the exonuclease activity as well. As of now, focus here. Now see, the point is very simple. No doubt this DNA polymerase 3, right? This DNA polymerase 3 is the main replicating enzyme, but still it is having the limitation. What limitation it has? It needs free 3' group for synthesizing the DNA strand. Okay, so it needs free 3' group for synthesizing the new strand of DNA. See students, it's very simple. That's your replication fork. You can imagine same thing for the replication bubble. The only difference is they are moving in the opposite direction. Now you know that here you have one strand having polarity 3' to 5', another strand having polarity 5' to 3'. Your helicase is moving like this. Your DNA is opening like this, in this way, in this way. Is that clear? Now, I know the process is going on in cytoplasm. I know even in the case of eukaryotes, I know the process is taking place in the nucleus. So there you are having everything required like DATP, DCTP, DGTP, DTTP. All that things are there, bache. All that things are there. These are what? These are your what? Nucleoside triphosphates, isn't it? These are, Subhashini, your nucleoside triphosphate, deoxyadenosine triphosphate, deoxycytosine triphosphate, deoxythymidine triphosphate. That's what we have. Now the point is, when I need to synthesize the new strand of DNA, I don't need TPs. I don't need triphosphates. I need monophosphates. I don't need DATP. I need DAMP. So these compounds, they are acting like the substrate as well. Why substrate? Because I'm going to use them for polymerizing or for synthesizing new strand. Plus, because they have 2 extra phosphates, so that phosphate will be released. That energy is used for this polymerizing activity. So it is acting like a substrate as well. It is acting like something which is providing the energy as well. Done, bache? Done, bache? Done, bache? Okay. Okay, this is given in NCERT. They have the dual function. They are acting like the substrate as well, and it is also providing the energy for the polymerizing activity. Okay, so now this DNA polymerase 3, everything is available. Everything is available in the vicinity of this DNA polymerase 3. It is having the substrate as well, right? It is itself is the enzyme. It is having the substrate. DNA strand is open, but still it cannot start the polymerization by its own. It cannot start a new strand synthesis by its own. It needs the free 3' group. Like let's say you cannot start some. Let's say you cannot start something by your own. Let's say you are cooking something. Okay, you don't know. You don't know how to start and everything. But if your mom is there, if your mom is guiding you and your mom is telling that I have done everything, you just have to put the salt and then stir it for a while. Okay, okay, right? You are functional in that way. Same is the case with the DNA polymerase. So this DNA polymerase 3, okay, it can synthesize DNA strand, but when it will see free 3-OH group. So now the point is, how can we arrange, right? How can we arrange that DNA polymerase? How can we arrange that free 3-OH group? Isn't it? Now this is the question. Am I right? This is the question. Okay, so DNA polymerase 3 is incapable of initiating the DNA synthesis by its own, right? It needs another enzyme for its help. Now there comes the role of primase. Which enzyme is going to play the role now? It is the primase. Clear, Subhashini? It is the primase. So primase, it forms RNA primer. And what is this RNA primer? It is near about 15 to 20 nucleotide long RNA chain. It is near about 15 to 20 nucleotide long RNA chain. RNA primer, right? Primase, that's an enzyme. It is DNA dependent enzyme. It needs the DNA template, and then it can form what? It can do what? It can form RNA primer. I'll give you one example. Let's say here you have C, you have G, right? You have T, okay? You have G. Let's say, imagine it. So obviously RNA primer, because it doesn't, primase, it doesn't need any free 3-OH group. So complementary to it, it will add the bases like G, like C, like A, like C, like this. Are you getting it? Like this. So primase is going to synthesize what? It is going to form the primer. And now see, let's say if it is a primer, obviously it will also having this thing. If it is a primer, yes, bache, if it is a primer, it is having this thing, 5' to 3' dash. Yes, bache. If it is the RNA primer is nothing, it's a stretch of RNA. That's how you can keep it in your mind. It is a stretch of RNA. Okay, so it is having the bases, it is having that nucleotides, it is having that nucleotide. So you know that, right? Whether it is DNA or RNA, 5' will have free phosphate group, 3' will have free OH group. Remember this part? 5' will have free phosphate group, 3' will have free OH group. So when primer will be synthesized here, right? When primer will be synthesized here, so immediately, right? Immediately your DNA polymerase will be like, wow. DNA polymerase 3 will be like, wow, I can see the free 3-OH here. I can see the free 3-OH here, and your DNA polymerase, right, will take over the role, and it will start synthesizing. It will start synthesizing the new strand of your DNA. Are you getting my point? Are you getting my point? So that's how your DNA polymerase 3, it works. Is that clear? First point clear, right? The role of primase clear. The role of primase clear, right? So DNA polymerase 3, when it will see free 3-OH group, it will come up and then it will further start adding the bases. Clear, bache, Shrutik? Sure. But now there is one more twist, right? Now there is one more twist. Now, bache, you know that when it comes to the DNA is a double-stranded structure. DNA is a double-stranded structure, isn't it? Yes, it's a double-stranded structure. Now, the story is DNA polymerase 3, right? DNA polymerase 3. It is moving like this. Fine. It has to synthesize the strand of DNA. It is moving like this. It is adding the bases. Like, let's say I'm moving like this. I'm the DNA polymerase. I'm adding the bases, right? This is how I'm moving. I'm adding the bases. But I have to do it in two, right? I have to do it in two different directions. What does it mean? I know my DNA is double-stranded. DNA polymerase 3 is moving like this very easily. It is adding the bases here, but it has to synthesize the another strand as well. It has to synthesize the another strand as well, and simultaneously it has to do. That is not the case. That is not the case, Trisha, that I'm going like this and then I'm coming back like this and then I'm synthesizing another strand. No, no, this is not going to happen here. Fine, this is not going to happen here, right? I have to move like this only. I have to do both the tasks simultaneously. Okay, I have to do both the tasks simultaneously. First point clear? Are you sure about it? This point clear? Okay, so now there is a problem. No doubt our DNA polymerase 3 is the main replicating enzyme, but it is having so many problems, right? It's very demanding. One thing is I need free 3-OH group, and second problem is DNA polymerase is like I can only synthesize in this direction. I can only synthesize in this direction. Effortlessly it can synthesize the new strand in 5' to 3' direction. Now look at it. This is one template strand having polarity 3' to 5'. That's how your application fork is moving, no? Right? That's how your application fork is moving. That's how your helicase is unbinding the DNA. So that's why it is the direction of your application fork. It is the direction of your application fork. Okay. Okay. So now what is happening? Look at this template strand. This template strand is having the polarity 3' to 5'. Yes or no? This template strand is having polarity 3' to 5'. Now look at this template strand. It is also having the polarity 5' to 3'. In this direction, because things are moving, so you have to consider the polarity accordingly. So this particular DNA strand is having polarity 3' to the 5'. This one is having the polarity 5' and the 3'. Is that clear? 5' and the 3'. Is that clear? Trisha, will you wait for some time? Let me finish the topic first. Okay, okay, so that point clear? That point clear? Yes or no? That point clear? This template strand is having polarity 3' to 5'. This one is having polarity 5' to 3'. Now my DNA polymerase is like, see, you know that they are anti-parallel strands. So here, the synthesis is very easy, 5' to 3'. It is very easy. Okay, it is very easy. So this strand is also known as your leading strand or the continuous strand. Or the continuous strand. This is the leading strand or the continuous strand. Clear, bache? Leading strand or the continuous strand it is. Continuous strand it is because continuously it is getting synthesized. Because DNA polymerase can move in this direction 5' to 3'. Now simultaneously it has to form a new strand here also. But here, bache, the synthesis, the direction of synthesis is opposite. Can you see that? Here, the direction of synthesis is opposite. Can you see that? Can you see that? Here, bache, 5' to 3' is the template. Okay. So, if complementary to it, if I have to synthesize the DNA, so the polarity will be like this. The direction of 5' to 3' will be like this. So, imagine, here, the scenario is, right, once DNA polymerase 3, it needs to synthesize one strand like this. And it has to synthesize another strand like this. Right? Imagine, it has to do something like this. It has to do something like this. Like it is going, one strand is synthesizing, it can synthesize in 5' to 3' direction. And parallelly, it has to synthesize another strand, which is in opposite direction. So that's how it is working. That's how it is working. Okay. Okay. Imagine I am the DNA polymerase 3. I am synthesizing 5' to 3'. Here also I am synthesizing 5' to 3' like this. So it is difficult for the DNA polymerase 3. It is difficult for the DNA polymerase 3. DNA polymerase 3 is like I can work efficiently in one direction. Right? If you will say that you have to form two strands, but in the same direction, then I will be very efficient. It's difficult for me. It's difficult for me. Because DNA polymerase 3 is like I have to coil the strand and then I have to synthesize it. Okay, you should check one animated video on the basis of that. You will understand it in a better way. So one strand synthesis is easy for me. I can do it like this effortlessly. See, I'm moving this hand effortlessly. But when it comes to like this, it will take some efforts. And they are in opposite direction. So here what is the thing? So polymerase is still giving its best Okay, polymerase is still giving its best. It is still synthesizing the strand, but here that strand synthesis is discontinuous. It is not continuous, it is discontinuous. Clear, bache? This strand synthesis is discontinuous. Like it's a patch of strand will form, then the connection of DNA polymerase 3 with that strand will break. Then another patch will synthesize, another patch like this, like this. Are you getting my point? Like this. And that's why it needs the polymerase, primase again and again. So again, primase will come, will form RNA primer, DNA polymerase 3 will form the strand. Then again, primase will come, again form, again DNA polymerase will come and strand will form. So that's how here the synthesis will be. That's how here the synthesis will be. So here one strand is continuously synthesized, leading strand. Another strand is discontinuously synthesized. So we also call it as lagging strand. What we used to call it? We used to call it as lagging strand. And bache, it was discovered by a scientist. The name of the scientist is Okazaki. Okazaki. So that's why these fragments, they are also known as Okazaki fragments. These fragments, they are also known as Okazaki fragments. Is that clear? These fragments, they are also known as Okazaki fragments. Is that clear? Sure, bache? Are you sure? No, it is not related to the mutation or something, Shrutik. It's just that because DNA polymerase 3 is moving in this direction. And yes, one student was typing it in the chat section. Yes, it is the direction of the replication fork. So this leading strand is moving towards this leading strand is synthesizing towards the direction of replication fork. These are the kind of questions that used to come in your exam. It is towards the direction of replication fork. And this one, this leading strand synthesis is, its synthesis is away from the replication fork or away from the direction of the replication fork. So that's what you need to keep in your mind, right? That's what you need to keep in your mind. So here there is only one RNA primer required and the complete strand will form. But here you need primers again and again, and that's how this lagging strand, this discontinuous strand will form. So you can see their direction. Yes, it is also moving in the direction of replication fork. It is moving away from the direction of the replication fork. That is the point here. So up to this part all clear? Up to this part all clear? So in the paper they can ask you, right, that leading strand form complementary to which template strand. So you should know about the polarities, student, right? You should know about the polarities. Yes, they can ask you that leading strand forms complementary to which strand, right? Which template strand. So you should know the template strand having polarity 3' to 5', complementary to it, leading strand will form. The template strand having polarity 5' to 3', complementary to it, lagging strand will form. So that's how you need to keep in your mind. That's how you need to proceed. Is that clear? Sure, students? Right? Up to this part all clear? Are you sure? So that's how the synthesis will occur. And what is going to be, what will be added here? Your monophosphates. So that triphosphates from the surrounding will be picked up. They will act like the substrate, plus they will release the energy for this polymerizing activity. Now I'll come to one point. So Bachit, synthesis is in 5' to 3' direction. Am I right? Synthesis is in 5' to 3' direction. Am I right? Right. Now, because you know that DNA replication is a very important process. If your DNA is not replicated properly, if it is not replicating properly, it can result in the mutations, and that mutations can be dangerous, right? So yes, there are certain checks on this particular mechanism, and that checks are even required, right? There are certain checks on this mechanism, and that checks are even required. So once the DNA synthesis is done, right, I told you we have 3' to 5' exonuclease activity enzymes. We have the enzymes which are having 3' to 5' exonuclease activity. I told you this thing. We have the enzymes which are having, right, 3' to 5' exonuclease activity. I told you. Now here comes the role of that enzymes. Right, bache, your DNA polymerase 1 and your DNA polymerase 2, they are the main enzymes which are playing role in the DNA repair and the damage. Okay, okay. So the first point that you need to understand here is this 3' to 5' exonuclease activity. Now imagine, let's say, imagine a process, right? You are making something, okay? You are making something in a proper direction. Let's say you're making something from point A to B and you have to cross check it. You have to cross check it. Let's say you are arranging your books from A to B and you have to cross check it. So what will you do? When you will reach at point B, you will come back like this. Okay, fine, let's check whether I'm making mistake or not. Sometimes we do such opposite countings as well, na? Sometimes we do such opposite countings as well, na? So let's say you have arranged something from point A to B, you will move from B to A and you will cross check it. Isn't it? Isn't it? Isn't it? Of course, same is the case here. That's how I remember it. Isn't it? That's how I remember it. So 3' to 5' exonuclease activity. 3' to 5' exonuclease activity, Nandini. So 3' to exonuclease means it is something which will cut the ends, right? It can cut the bases from the ends, from the terminal ends. So this enzyme, it will move from 3' to 5' and it will make sure that all the bases are added properly or not. Okay. Okay, this is the role of this 3' to 5' exonuclease activity. It is going to make sure that, right, it will act like a check system. It will check, right? It will move from 3' to 5'. Why do we say 3' to 5' exonuclease activity? Because this enzyme will move from 3' towards the 5' side and it will keep on checking that all the bases are added properly or not. Right? All the bases are added properly or not. That's how it works. So understood, bache? Sure, bache, Shri Chandana? Is it understood? Is it clear? Is it clear? Are you sure, Vidisha, everyone here? Are you sure? Okay, 1 point. Now, I also told you that DNA polymerase 1 is kind of unique, right? This is something very unique to your DNA polymerase 1. That's very important. Please listen to me carefully. DNA polymerase 1 is unique because it is having this activity. Actually, your DNA polymerase 1, it plays a role in gap filling as well and primer removal as well. And primer removal as well. It is playing role in gap filling as well and in primer removal as well. Now, Trisha was asking one question. Trisha was like, ma'am, when you are making the RNA primer, RNA is having the uracil at the place of thymine. Trisha, are you there in the class? Now you will get the answer of your question. Are you there in the class? So Trisha was asking one question that, ma'am, RNA primer is having the uracil at the place of thymine. RNA primer is having the uracil at the place of thymine. Right? Why is it so? Why is it so? Why things are like that? DNA doesn't require, right? There is no need of uracil in DNA. But if RNA primer is here, right, then it will contain uracil. Then how can we say that our DNA is perfectly fine or something? Isn't it? So that's why, bache, that's why we have to remove, we have to remove these primers. Once the DNA synthesis is done, once this part is done, you know what is going to happen? You know what is going to happen? This primer will be removed. This primer will be removed. This primer will be removed. Are you getting my point? Are you getting my point? Once, bache, please check my previous lectures. You will get the answer of your question. In the nucleosome coiling, we have discussed that. Are you getting my point? So that primer will be removed. So this 5' to 3' exonuclease activity, which is something unique, what will it do? It will remove the primer. That's very important. It will remove the RNA primer, right? That's unique. MCQ can come from this part, right? This is something unique related to your DNA polymerase 1. So that's what you need to keep in your mind, students. Fine, that's what you need to keep in your mind, Aarti. So it will remove the RNA primer. Now look at it. So if I will remove the RNA primer here, if I will remove the RNA primer here, don't you think there will be the gaps? Don't you think there will be the gaps? Imagine. let's say this is your lagging strand. Fine. Let's say this is your what? This is your lagging strand. Which strand it is, students? Which strand it is? It is your lagging strand. So lagging strand is having these so many RNA primers. Leading strand is having only one RNA primer. Lagging strand is having many RNA primer. So your DNA polymerase 1 will remove them. It will remove them. So don't you think the gaps will come in between? Don't you think that gaps will come in between if RNA primer will be removed by DNA polymerase 1? And 5' to 3' exonuclease activity is going to do the same. So don't you think it is going to happen? Of course. Of course. So for this gap filling, again your DNA polymerase 1 will play its role. Okay. The primer removal, no Nandini, there is no role of ligase yet, bacha. Right? So for the gap filling also, for the primer removal also, DNA polymerase 1 is going to act. Right? It is going to work here. So I told you that even the DNA polymerase 1 is having 5' to 3' polymerase activity. I told you this thing. Even it is having 5' to 3' polymerase activity. So it will add the DNA bases here. It will add that deoxyribonucleoside monophosphates here. Are you getting this? So this gap filling will be done by your DNA polymerase 1. And then Nandini, there comes the role of your DNA ligase. Then comes the role of your DNA ligase, which is also considered as molecular glue. So once these bases are added here by DNA polymerase 1, so that sugar phosphate backbone will be attached. It will be joined with the help of DNA ligase. It will be joined with the help of DNA ligase. So DNA ligase is going to do what? DNA ligase, it is a kind of glue. So it helps in ligating the sugar phosphate backbone. Okay, it joins that sugar phosphate backbone. Clear, bache? It joins the sugar phosphate backbone. Is that clear? Is that clear? So that's the wonderful role of your DNA polymerase 1. It is even going to remove the primer with the help of its 5' to 3' exonuclease activity, plus it will fill the gaps with that 5' to 3' polymerizing activity. So that's how this process occurs. This is all about your DNA, right? DNA replication. Clear, bache? Clear, bache? So discontinuous pieces of lagging strands are joined together by the enzyme DNA ligase. Is that clear? Is that clear? Your DNA ligase will join them further. Right? Right? So you can simply keep it in your mind that DNA ligase will further join these breaking strands. These gaps will be filled by your DNA, joined by your DNA ligase. Is that clear, Sabha? Sure? 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 Okay, so this is the role of your DNA polymerase 1. And let me tell you, DNA polymerase 1, you know that it is playing role in the repair as well. So sometimes because of mutations, because of UV rays, thymidine dimers form in DNA. Do you know that? Do you know that? Because of mutations, because of ultraviolet radiations, thymidine dimers, they form in the DNA. And your DNA polymerase 1 will break that, will fix that. So that's why it is also the repair enzyme. Okay, Arthi? That's why it is also the repair enzyme. So this is all about your DNA replication. Bache, we have covered each and every important point of that DNA replication. You are not going to get anything beyond this part. And I will show you the wonderful diagrams as well, so that you can understand this process, so that still you can recall it. So still, if there is any doubt, do let me know, then we will continue this. Quick, if there is any doubt, do let me know, and then I'm going to continue this part. Quick, waiting for your doubts. Quick. Sure? Sure. So let's quickly revise it from NCERT. Let's quickly revise it from NCERT. So here you guys can see the machinery and the enzymes first. Okay, so bachcho, how to revise this DNA replication? Yes, how to revise the DNA replication? Simply you need to keep one thing in your mind. Okay, simply you need to keep one thing in your mind, right? Just imagine it like a story, that there is the interface, right? And in the interface, the things are going like that, right? DNA is opening with the help of helicase. That's how it is moving. Okay, this is what you need to keep in your mind. Clear, bache? So, in living cells such as E. coli, the process of replication requires a set of catalysts. Okay, so the main enzyme here is DNA dependent DNA polymerase. So sometimes in the paper they confuse you. Sometimes they will write RNA dependent DNA polymerase or DNA dependent RNA polymerase. Here this DNA dependent DNA polymerase. Why DNA dependent? Because it needs that template, and then complementary to it, it will form the new basis. Fine, bache. So since, see, the reason is also explained. The main enzyme is DNA dependent DNA polymerase because it uses a DNA template to catalyze the polymerization of deoxynucleotides. So you know that this enzyme is highly efficient, right? So and they have to catalyze polymerization of large number of nucleotides, right? So you know that when it comes to the E. coli, it has 4.6 into 10 raised to power 6 base pairs, and in humans it is 6.6 into 10 raised to power 9 base pairs. You need to remember this data. Okay, you need to remember this data, bache. Okay, thank you, Aarti. Thank you so much, bacha. So it completes the process of replication within 18 minutes. I told you in the E. coli 20 minutes is the generation time. So this replication process will be completed in 18 minutes. We have to follow the NCERT here. But if you will study some foreign author books, you are going to find 20 minutes there. Okay, so average rate of polymerization has to be approximately 2000 base pair per second, and trust me, DNA polymerase 3 works in that way, works in that speed. Clear, bache? So it is not just about being fast, but it has to be accurate as well. Just in the final NEET paper, right? In the final NEET examination, guys, in the final NEET examination, you know that you need to be fast. You need to answer questions quickly, but you need to be accurate as well. Right? You need to be accurate as well. Are you getting it? Once you need to be accurate as well. Why accuracy is important? Otherwise, you know that there will be the negative marking. So yes, by once, bache, 38 minutes is given for one-sided. But in E. coli, it's bidirectional. So in totality, we consider, bache. So any mistake during this DNA replication will result in mutations. So it is, see, energetically replication is very expensive. So one student was saying, no, I don't know about this point. See, so deoxyribonucleoside triphosphate, they serve dual purpose. They are acting like a substrate as well, and they provide energy for polymerization reaction as well. So please take a note of it. Take a note of it. It is important. It is important. Done, bache? It is important. Clear, bache? So in addition to DNA dependent DNA polymerase, many other enzymes are required, right, bache? So 2 strands of DNA cannot be separated in its entire length, right? Right. So due to very high energy requirement, the replication occurs within a small opening of the DNA helix, which is known as replication fork. So Y-shaped replication fork is there. So DNA dependent DNA polymerase, it catalyzes polymerization only in one direction. You know that, 5' to 3'. That's why when it comes to the synthesis of the strand, which is opposite to it, it is difficult. That's why we get the lagging strand there. Right, Subhashini? So see, this creates some additional complications at the replication fork. Consequently, on one strand, the template having polarity 3' to 5', the replication is continuous. While on another strand, where the template is having polarity 5' to 3', it is discontinuous. Another important MCQ. Okay, it is another important MCQ you need to remember. So when the template strand is having polarity 3' to 5', complementary to it, leading strand will form. When the template strand is having polarity 5' to 3', then complementary to it, lagging strand will form. That's what you need to keep in your mind. So discontinuously synthesized fragments are later joined by the enzyme DNA ligase. So it joins the sugar phosphate backbone. Okay, it joins the sugar phosphate backbone. So DNA polymerase on their own cannot initiate the process of replication. So replication doesn't initiate randomly. You know that it will start from the origin of replication. So how many origin of replication are there in prokaryotes, student? How many origin of replications are there in prokaryotes? How many origin of replication are there in prokaryotes? Quick. Only one. But in the case of eukaryotes, there will be many. So we used to say polyreplicate. Okay? Done? Done. So, because of the requirement of a region of replication, that a piece of DNA, if needed to be propagated during rDNA procedure, it requires a vector. But it is related to biotech, right? We will see the role of these vectors. We will see the role that how, this origin of replication is helpful for propagating some another genes as well. Fine. So not every detail of replication is understood well. So in eukaryotes, replication of DNA takes place at S phase. So that's something very important. Right? I was waiting for this point. Students, when it comes to the prokaryotes, initially I told you now that replication occurs in S phase, but not in the case of prokaryotes. In the case of bacteria, you do not have that proper meiosis, mitosis, you know it. They do not have this interphase and M phase. No, it will be there in the case of eukaryotes only. And in the case of eukaryotes, you know that it is not DNA polymerase 3. In eukaryotes, DNA polymerase alpha, beta, delta, epsilon. There are different type of DNA polymerases. Okay. So right bache. So if there is failure in the cell division after DNA replication, it results into the polyploid chromosome anomaly. Right? If this DNA replication is not proper, if later on in the daughter cell, if it will not get separated properly, polyploidy can occur, and it's very common in the case of plants. Fine, it's very common in the case of plants. So let's have a look of this. So diagram is important. You need to even focus here. So this template strand is having polarity 3' to 5', right? Leading strand is formed. And here it is discontinuous, 5' to 3'. Lagging strand is formed. Clear, bache? So look at this diagram. So see, this is how your helicase is moving. So helicase is moving like this. Your replication fork is moving like this, right? And look at this leading strand, because it is also moving in the same direction. So see, synthesize. This synthesis is continuous. So this is basically the example of your eukaryotic. DNA replication. Why? Because of this thing. Here the DNA polymerase delta is there. Okay, DNA polymerase delta is the main replicating enzyme there. And now look at this. This strand synthesis is in opposite direction. Can you see that? Right? So that's how it is moving. So see, DNA primase is there. Okay, the primase is there. These are single-stranded binding protein which will not let them to join again. Right, bache? This is topoisomerase which will remove the supercoilings. Okay, so see this diagram also. So that is your replication bubble. So replication bubble is having two replication forks. Okay, it is having two replication forks. So look at the leading strand continuously. Look at the lagging strand. So lagging strand, it is moving away from the direction of the replication fork. So see, primer, the lagging strand. And here, this is your DNA polymerase 1, which will remove the primer and will play a role in the gap filling as well. And how will it remove the primer? With the help of this 5' to 3' exonuclease activity. Is that clear? With the help of this 5' to 3' exonuclease activity. Is that clear? So look at that diagrams. Okay. So that's the summary. Even here you have the summary as well. Helicase, primase, right? Single-stranded binding protein, topoisomerases, gyrases, ligases, primases. So these are the enzymes. So I'll provide you the notes in our official Telegram group. You can get the idea from there. Fine, you can get the idea from there. So this is all about your DNA replication. So guys, if there is any doubt, do let me know, because the next topic that we need to start is transcription. Okay, the next topic that we need to start is transcription, which is also very interesting, right? You remember the central dogma, DNA to mRNA and mRNA to protein. That's how the information flows. So transcription, and then comes the translation. So you already know about the replication. Now what we have to study? Transcription and then the translation. Is that clear? Then the translation. So now, bache, it's a lengthy topic, so we will start it in next class. I know, in the morning slot we are not getting that time, so I will not for this week, but from next week I'll try to take that morning sessions for 2 to 3 hours. Is that okay for you? Is that okay for you? So I will try to take these morning sessions for 2 to 3 hours, so that in 3 parts or 4 parts we finish this. Better now? So in one flow we will finish it, right? So same way the marathons will be there in the evening, right? I will just increase the frequency and I will increase the duration of the class. Frequency and duration, both I will increase. Fine. So now you know what you have to do. Just let me know in the comment section that you liked the video or not, right? Do you like the session or not? And if you're new, you have to subscribe our channel, right? So I'll send all the PDFs together. Fine. I'll send all the PDFs together. Okay. Done? So if there is any doubt, do let me know. Sure? Take the session at 6 a.m. AM nahi at 2 p.m. is fine, Soumyadeep. 6 a.m. Bache, Glacier, you can be the part of Conquer batch. It is going to be the best for you. So I'll wait for your comments in the comment section. So stay tuned and take care. See you all tomorrow at 10:30. And yes, from the coming week, I will increase the duration of this part as well. Right? I'll shift my Avengers batch class. I'll shift that class in the evening and then I will discuss this with you. Fine? Okay. So thank you so much. Take care. Stay tuned.